Andreas is first a linguist, then a neuroscientist, then a neurolinguist, now a cognitive scientist, assistant professor, and upcoming decision-making teacher / researcher. So Andreas can do a lot, and he's always got very interesting things to say. So thank you, Andreas, for giving us, is it the last talk of the year, I think? Yeah.
I should have brought jingles and...
One of my least favourite Danish expressions, but the raisin in the end of the sausage. Andreas.
Thank you, Josh. All right, thanks for having me. This is a bit of work in progress, so hopefully it can lead to sort of a little bit of inspiration. It's probably a little bit of scattered thoughts here and there. But let's see how we go. Yeah, cool. So I put the title as Cognitive Adverse Effects of Parkinson's Disease and its Treatments. And I think it's mainly going to be treatments, because that's where my main focus is, but it's going to be a little bit of a mix of both still. Okay, cool. So plan for the talk is that I first will tell you a little bit about Parkinson's disease so that we're on the same page. And then I'll get into the cognitive adverse effects of the disease and the treatments that I'm particularly interested in. Okay, so this will mainly be language related, and this will be verbal fluency, and then action language, in lack of a better word for that. And then I don't think we'll get to impulsivity, so I'll just mention that at the end. That's at least. So I'm aiming for 45 minutes. Right, Carsten? Good, excellent. Okay, so. Thank you for the very nice introduction, Josh. And then this is in writing, what Josh just said. So I'm not going to spend more time on that, because what I want to do, because there are some familiar faces here, also some, of course, new faces, is that always start on a happy note. And this is completely unrelated to this, but this is a magnificent feat. This, yes, for those of you familiar with my history. 10 years ago, in November 2012, so it began. They sent out a calling for, almost, okay. Anyways, but you see Mikkel's name is on here. Mikkel and I, we packed down the entire EEG lab of Sefin and drove it across the snowy countryside in a van all the way to Copenhagen. We found in the outskirts of Copenhagen the barracks of the language officer academy of the Danish military. And we subjected them to proper neuroscience 4 times over the course of 2 years. We were drenched in sweat after packing down and re-unpacking the EEG lab 4 times and setting it up in various crazy barracks locations in Copenhagen. And then finally, after many tears and cries, I submitted my PhD and it was rejected and I had to redo everything again. Just because of this study, where we found absolutely no effect of learning a foreign language on the neural responses to the sounds of those foreign languages. But the study was so beautiful. And then finally, 10 years later, New Image Reports also thought so. Well, actually they didn't. But I just kept nagging them and nagging them and nagging them, and then at the end, the editor of the special issue, which is called Null Results, very adequately named, was so tired of me because it was a year since the special issue had actually properly ended. She finally said, okay, the reviewers don't agree, but let's publish it anyway. Anyway, so that's a very happy note. Yes! It's mainly to say it's actually a really, really neat study. It's just, it suffers under no really interesting results in it. So I'm just really happy that it's out there and documented. It's been out there as a preprint for long, but this makes it a little bit more official so that people who could just by chance happen to think, okay, mismatch negativity, could that be an index of language learning? Ah, that would be really fascinating, interesting to do. No. Just don't. There's no reason to do it. I've done it. It took us, 3-4 years and then 10 years to publish. There's no reason to go there. Okay, cool. I normally also start by showing what I look like when I do research. here is me. And then of course it's all the very skilled and excellent research assistants that actually do the proper work. But this is from the barracks, so we actually did it. This is the language officer cadets sitting here getting prepped with EEG, and then me pretending to look important and saying something to them that they engage with. Anyways, that was sort of a nice wrap up on me and my history. So, that big endeavor actually led me to into Parkinson's disease. There's a very clear line from that to that. It makes perfect sense in my head at least. Of course, the methods that I applied during my PhD, mainly EEG and MEG, in sort of a cognitive neuroscience, neurolinguistics framework, was actually very sought after. And so the very small group here at Aarhus University / Aarhus University Hospital, looking at Parkinson's disease patients, and in particular the deep brain stimulation treatment, we're actually looking for someone to help them work with that. And that's then how I got into Parkinson's disease. Yes. So that's the segue into that. And that's what we will be talking about today after we landed on that happy note. I'll take a little bit of water. Okay. So Parkinson's disease is not nice. Really it isn't. It's one of the diseases that we call neurodegenerative. So that just means that the brain just gets worse and worse. So that's really crappy. Furthermore, we don't really have anything that fixes it. So the treatments that we have are symptom reliefs. So most of the time PD patients are treated with medication, levodopa in particular, which is sort of like a precursor for the dopamine that they're lacking, that they don't have in the brain. And so that works for a couple of years. For some it lasts for 7-10 years, for some it only lasts 3 to 4 years. There's a lot of variety in that. Once they then start having too many side effects of the medication compared to the symptom relief that it gives them, there are other advanced treatments available. And one of those is called deep brain stimulation. And it's actually probably the method, the treatment where we stimulate the brain electrically that works the best amongst those that are out there. So the very few cases where we actually put electrodes deep into people's brains and stimulate those brains electrically, this is the disease for which it works the best, and probably has also been around the longest. Yes. So again, this is a busy slide, but we don't know too much about exactly how Parkinson's disease starts. There are hypotheses out there that either it starts in the gut or it starts in the brain. Anyways, it's a protein called alpha-synuclein that misfolds. It does that for all of you. You have garbage in your brain right now, and your brain, for your body, it's also in the body, fixes that. That's great. For PD patients, those proteins misfold too much, and they sort of clot up, and then they lead to cell death, right? And that's why it's a neurodegenerative disease, and this particularly targets the dopamine-producing neurons in the midbrain. And we use dopamine for a lot of things, but where for PD patients this is most detrimental is within the basal ganglia, deep inside the brain, helping them regulate their motor, the movements in general. Okay? So what happens is that you often have a sort of prodromal stage before you can actually diagnose PD, so 5, 10, maybe up to 15, 20 years, where this could sort of be in the making. And then not until you have sort of clear motor symptoms can you diagnose the disease. And at that stage, most patients already have lost about 50% of their dopamine-producing neurons in the midbrain, right? So we're already pretty advanced into the disease. This is, of course, a sort of a broad generalization, and it does vary a lot from patient to patient. These are the sort of the cardinal symptoms of it. You have bradykinesia, which is slowness of movement, right? So they generally just get very sort of erratic and slow in their movements. Then you have rigidity, they get very rigid in their muscles and in their, And then for a subgroup of them, they also have tremor, resting tremor. Not all of them at all, but definitely a lot of them have resting tremor. And this is not to be confused with what's called central tremor, familiær rysten in Danish. Some of you may have heard a lot about that very recently, because at Skyby, they're right now actually burning away a little bit of the brain for those, for that disease. And that is tremor whenever you have a sort of goal-directed movement. So it's a related disease, but it's not the same. So, as I said, it's initially treated with meds, and then later on, this medical treatment or medicine treatment leads to many so-called off stages. That is where they're treated with the medication, but they don't have an effect of it. We can talk about the mechanisms of that if that's interesting, but we're not going to spend more time on that, except for the fact that that's when they become eligible for this deep brain stimulation treatment. Okay. Generally speaking, the deep brain stimulation treatment works great for the motor symptoms, really. Like, it really sort of brings them back several years in terms of their motor function, and it has rather little, rather few side effects. However, for a few of the patients, we do see some cognitive adverse effects. This mainly has to do with language related, or at least sort of language/executive functioning. It's not super clear, but what is most often reported is what we call verbal fluency and then impulsivity, and I'll get back to that in a second. So, just to be clear, I mentioned basal ganglia. This is sort of a schematic drawing of the basal ganglia, which are hiding here deep inside the midbrain, and they are essential for a lot of what happens right now in what you do. One of the things that they are really good at is coordinating movement. And it's an intricate network of connections. So there is both a direct and indirect pathway that connects all of these different small nuclei, leading to one particular central nuclei, namely the globus pallidus internus, which is the only output nuclei of this whole sort of basal ganglia system, which feeds directly into thalamus, right? This is an inhibitory connection. So generally speaking, we can think of the basal ganglia as a very, very advanced brake that you sort of can control, and then the degree to which you sort of increase the output from this one, the more you sort of put a hold on everything else. It's a gross oversimplification, but it kind of works, okay? And this kind of also makes sense in terms of how PD symptoms sort of look, because they have this rigid bodily function. They also have these erratic movements where they can't really sort of engage in an activity. So this is when this activity is, there's a burst of unregulated activity in here that heavily inhibits anything that passes through thalamus. Okay? Yeah. So most of what the medical treatment and so on targets this whole system with balancing the dopaminergic levels in here. What then happens is there is actually sort of an extra level to this network, because as you can see, we have the connection back up to cortex, which is functioning just fine. In PD patients. So, for instance, when they have this symptom called freezing of gait, where they when they meet an obstacle, for instance, walking through a doorway. Yes.
That was an obstacle.
There was a true obstacle. This is almost an imaginary one, but not to the PD patients. They will get stuck here. Simply just stuck. They can't move. But what some of them can do is count themselves out of it. Højre, venstre, højre, venstre, højre, venstre, højre, venstre. And then they can actually sort of bypass the automatic basal ganglia function via the sort of cognitive motor cortex instructing themselves to move. Okay? Just to say that there is some part of this network that is still relatively well functioning. And that's relevant because there is a hyperdirect pathway from cortex projecting right onto this tiny nucleus down here called subthalamic nucleus, which heavily impacts GPI. So when the medication stopped working for the treatment, and we want to place an electrode in this system or network to sort of help regulate matters, oftentimes it would be targeted at GPI, right? Because then you stop things at the output and sort of regulate things there. But it seems as if targeting subthalamic nucleus has the same effect and is a little bit easier to hit, you could say, because you have to put the electrodes down through large parts of brain tissue without damaging anything. And also suggestively maybe have a little bit of better effect on the medication that's needed. Okay, so that's also a nice thing, that you can draw back some of the medication and get rid of some of the side effects of that. Okay, cool. What is curious about that is that even though these projections come from cortex onto subthalamic nucleus, when we stimulate it electrically, those connections can actually be stimulated, what we call antidromically. So normally a neuron can only shoot an action potential out through its axon. But when you stimulate, because that's an electrochemical process, but when you stimulate electrically, you can actually have activity activated back up in cortex through that electrical stimulation of that particular neuron, those projections. Okay? So that will all sort of become relevant in a sense, because this is the big picture now, that when we implant these electrodes down into subthalamic nucleus down in here, we have a few different contacts we can stimulate on to optimize the treatment. And then normally what is targeted, because this is purely from like a motor symptoms perspective, is the dorsolateral region of it, so towards the back and towards the side of this tiny little almond-like structure, so that you hit the projections to motor cortex and into the motor part of GPI as best as possible. And from a lot of sort of old animal literature, it was thought that there was this sort of tripartite division of the nucleus, subthalamic nucleus, but then recent literature suggests that it's a much more sort of mixed bag of things, right? So you have projections to, for instance, more prefrontal cortex, also mixed in with some of that dorsolateral region. And this will also be relevant in just a second, okay? Because that's how you can then alleviate stimulating electrically the motor symptoms of these PD patients. Now, as I said, this works for the majority, really well for the majority of patients, but we do see some cognitive adverse effects. And in particular, we see it for what's called verbal fluency, okay? So, impaired verbal fluency is seen for PD patients without stimulation, but then after deep brain stimulation, we see a worsening of verbal fluency. And what is verbal fluency? Verbal fluency is a task that's used a lot in the clinic. You have to list as many words as you can within that either begins with a given letter, for instance F, or within a category, for instance animal, and you have 1 minute to do it. And you can't mention any proper names, and you can't do sort of inflections or derivations. So if you have swim, swimmer, swam, so on, so on. It has to be sort of new words in a sense, and no repetitions. Okay? This is a really, really good task clinically. Because if you have issues, executive functioning, language related, this will most likely catch it. It's a really, really crappy task experimentally because we have little to no control over what happens with the patients. They just sit there for a minute and then we have to figure out what they did, right? Because what does it require to solve this task? Well, we call it cognitive flexibility. So that will, for lack of better words, so that relies both on some executive functioning and then also on some lexical search. You have to keep track of what you just said. You have to sort of make sure you don't mention these proper names that maybe pop up if you were mentioning things with F, or other things that could be improper to mention at that given setting. Furthermore, once you have sort of exhausted, you could say, a search space, if you're doing animals, maybe domestic animals, you have to sort of say, should I keep searching here and for new animals, or should I go to the savannah or whatever way you want to solve this? And we don't really have a lot of control over that. So the different cognitive processes involved: lexical search, memory retrieval, executive functioning, response monitoring, and inhibition, and also selection, right? So, because it relies on that many different cognitive processes, it's a really good measure in the clinic. But then when we have to figure out why PD patients have issues with this and even get worse after DBS, we're at a bit of a sort of a conundrum, where to go from here, okay? Because if you get a head injury, or if you have other issues with the head, you will most likely also show up on this global measure. So we want to at least hypothesize that there's something slightly particular to the PD patients, and we want to try to tap into that, okay? And that's why it's relevant to come back to this image. When we stimulate down here, we're most likely, at least for some of the patients, also stimulating their prefrontal cortex, maybe to a lesser extent the temporal lobes where they do the lexical search. So it may be more related to some executive functioning, is at least a hypothesis we could pose. And this is where the great work of Mikkel Petersen comes in, because we actually have these PD patients in the MR for an extensive period of time before they have their electrodes implanted. So we can do tractography on these patients. That's where we actually do fiber tracking of the connections. And so what Mikkel tries to do is he tries to map on the individual patient the projections from prefrontal cortex and other regions of the brain onto the STN. And then we try to compare that sort of STN profile of the individual patient with where, for this individual patient, the electrode is actually placed and where we're stimulating, right? So that we can sort of get a map of whether this particular patient has more sort of stimulation of, let's say, prefrontal cortex. Because what we found out when we looked through the literature on verbal fluency is that this is one of the most reported cognitive adverse effects, especially worsening after DBS. Why is that? Most likely because it's one of the methods that you can apply the most, the easiest in the clinic, right? So that of course goes together. It should also have a deficit on it, of course. But what we also found was a high degree of individual variation in this. So it's reported as a group effect normally when we see it, but that is definitely not the case in the sense that it's an overall group effect. It's most often in the literature somewhere between 10 and 25% of the patients that show a clear worsening of the DBS, and the others show improvements or no difference at all, right? And that, of course, is what led us to look at the neuroanatomy and the fiber tracking coupled with where we stimulate to look at that. So that's what our sort of current project is undertaking. We're trying to stimulate at different places of the nucleus so that we can try to maybe even invoke a little bit of verbal fluency difficulty in these patients depending on exactly what part of the STN profile that we're stimulating. So that's still ongoing work, but we're still stuck with the issue that we can only sort of get a global measure of the verbal fluency performance. And that's where some of the work that I've done with a Norwegian psychologist, Einar, comes in, because through the years, these verbal fluency tasks, because they're so popular in the clinic, of course have undergone scrutiny in terms of how can we disentangle the processes involved in it, right? So one of the approaches that I came across is it's actually some really old literature. It goes back to the 70s. So if you have a lot of people producing as many words as they can for a minute or 2 minutes, the production rate will sort of decay over time. That's not very surprising at all. And it sounds kind of sensible in a sense, right? But then what you can do is if you fit sort of a decay curve to that, then you get sort of the expected production rate at any point of time in this process. And then what you will see is that the different words tend to cluster in smaller units, right? And this also goes together with the kind of, you could almost say qualitative analysis that has been done by neuropsychologists, especially by a guy called Tröster, who simply thought, well, if I can come up with a bunch of semantic categorizations of animals, then that will be applicable to all patients doing this task. That seems like a really sensible thing, especially with what we know about semantic categories. Anyways, that's how they did it, okay? So that's actually the golden standard. Tröster's sort of groupings of these words into different domestic animals, animals from the sea, animals that fly, whatever, something like that. Of course, it's very North American centered because that's where Tröster lives. Anyways, that's how currently this verbal fluency task is analyzed in a sort of decompositional manner, where you try to identify small semantic clusters and then switches between these clusters in sort of trying to mimic this lexical search process that we assume is going on, right? That's why I said it before, that these patients, we assume that they're trying to sort of explore a sub-semantic space, and then once they're done with that, then at some point they decide to move on, right? And that's the hypothesis that's is being sort of put forth with these deep brain stimulation effects on PD patients, that they have difficulties switching out from a search space. So what you see, at least as is reported, is that these PD patients, especially after DBS treatment, is they get stuck. And it kind of mimics what they also do motor-wise, right? They get stuck somewhere. So that's why we get a smaller production by them, that you see a pretty sort of normal rate. At some point they produce maybe 5 or 7 words, and then at some point they just stop for maybe 30 seconds. And then maybe they get out of that sort of stuckness, or they don't. And that's why their production is lower, okay? So that is what we're trying to target, but that of course heavily assumes that that's exactly how they do the task, okay? And having worked with recordings of these PD patients, automatizing this process is really not going so well, mainly because of bad auditory quality. That's unfortunately something we have to live with, because we're recording them while they're sitting in the scanner. And the data that I have together with Aya is also some pretty sort of standard psychology from a neuropsychologist, and it's just some really crappy recordings. So it's very hard to get the onset of the words properly, especially because there's also a lot of ers. And nobody talks about this in the literature, how to handle the ers in this. So I'm still working, and if you have inputs on it, I would happily take it in terms of sort of targeting, because we need these onsets. They're pretty crucial in terms of fitting this properly and thereby identifying the clusters. Also, for the cases where it does work and we have cleaned the data, the recordings really well, these clusters don't overlap very well with the semantic analysis. So that puts a little bit of pressure on that correspondence.
So these clusters, are they temporal only, or?
This is purely temporal.
Okay.
There's nothing to it but temporal.
Okay.
Yeah. So this is data from a patient. You can see how it says ventral and dorsal, so that means we have 2 different stimulation sites. And then we also have a site, a stimulation where we don't stimulate at all. And so we can sort of identify something that looks like very small clusters and some switches, but when we then look at the semantics of it, it really doesn't sort of form any sensible semantic clusters at all.
According to
Tröster's? Yes. Yeah.
Okay.
So you're segueing. Thank you, Carsten. Because should we necessarily then rely on Tröster? Do we have anything else we can do? And so I came across this one, computerized analysis of verbal fluency, done on English. And so it uses something called explicit semantic analysis, okay? And so what that is, it's scraping Wikipedia, specifically Wikipedia, using the already predefined categories there to then form a semantic network of relevant categories. This works really well for animals. And so we were quite excited about this, and Ira tried to use this for her master's thesis, comparing the method that I proposed to her with the temporal, not getting any good overlaps at all. We finally got the code to work, and then we figured, let's do this for Danish. And Finn over at DTU has actually set up a sort of a nice Python package doing ESA in Danish. And then it turns out that in order to get this to work, they simply fiddled around with different thresholds for when a semantic association between words should count as now high enough to be within a category and low enough to then be a switch. Fiddle around with different thresholds, 0.5, 0.75, until they got something that looked relatively similar to what they would get from a Tröster analysis. And then that would be the threshold that worked, and was the threshold that should be used for all eternity, at least, without doing more to that. And so we tried, of course, with Danish, also tried fiddling with the threshold, never got anything sensible at all. It's extremely sensitive, of course, to the data, you put into your ESA, your explicit semantic analysis. The English Wikipedia is ginormously bigger than the Danish, and so on and so on. So those weights are, yeah, we tried relative weights, we tried everything. And then, I mean, to begin with, it didn't make a lot of sense that we just had to find a threshold like that. Having fiddled around with it, it made no sense at all. So luckily, I have my research semester next semester, and that is what I will work on, trying to get this ESA to work somehow. And I'm very open to inputs as to how we could work with these weights to define clusters in a meaningful manner. If we should just do K-clustering, or yeah. So I'm very open to inputs on that, and I will come here for one of those Wednesday sessions and present it, so we can work more closely with it. So that's where we are right now, because I have the temporal analysis that I can use for the neurodata and so on, but I would really like to couple it with the semantic analysis as well. Yeah, Christian?
Yes. So rather than clustering, you could also just, I guess, directly correlate semantic distances and temporal distances, right? But why establish, these kind of, why is it necessary to establish clusters?
So why is it necessary to establish the cluster semantically? Is that what you're asking?
Yeah, because you could just use a continuous measure of semantic distance, couldn't you? So the idea is that you spend shorter time moving between two animals that are closer associated.
Yes.
Right. So I guess you could do the same analysis.
Yeah.
Without clustering.
Yes. Okay. So one thing I didn't mention very clearly is, of course, then that what we want to then look at when it comes neurally are what we could call good switches and bad switches. So we want those switches between the categories and compared to the ones within, and then to see if we can, for these PD patients, find the switches that are sort of abnormally long. And then, because the problem is, of course, that they're producing words when they're not switching. I mean, so when they're producing words, they have a lot of facial muscles activated, which makes the neuroimaging impossible to sort of work with, right? So we want to look at the switches. It's also where we think the processes that are relevant are happening. So for now, we can do the temporal analysis like this, and then we don't need the semantic per se, but having looked at the clusters that come out of this temporal analysis, with respect to, for instance, a Tröster analysis, which is not all crap, it does make sense to some extent, there's no sort of clear semantic overlap, at least. And therefore, I'm thinking they're not necessarily tapping into exactly the same kind of process, or there are two aspects of this process that I would like to disentangle.
Yeah, okay. But at first, kind of proof of concept could be to see if there's any correlation between semantic distance and
temporal distance. Absolutely, that's a good point.
But maybe
That's, but that's but that's a really good point. So that whether we all can use the weights and that's a really good point. That I can apply very readily. Josh.
On that, but also have a quick question.
Okay, cool. Yeah, sorry. Yeah, cool. Okay, because then that's the verbal fluency chapter. More generally speaking, language related. I came across some embodiment literature on the PD patients. The claim being that when they have difficulty moving about, then they may also have difficulties understanding action language. Apparently, especially the South American PD patients have this. There's at least one group that that insist on producing a lot of papers on this, showing PD patients single words, action verbs, words versus not action verbs, and so on, lexical decision task, picture, word matching, and so on, and find some sort of relatively, at least somewhat stable results. So I came across this study where they had the PD patients in Spanish read short stories, either with sort of a lot of action verbs or not so many action verbs in them. And then right after that they had to answer questions either about where the study took place or other sort of information about other circumstances compared to then questions that targeted actual action verbs. And then what they found was that for these PD patients that were not mildly cognitively impaired, which unfortunately also happens for a lot of PD patients, so they were still, in a sense, otherwise normally functioning cognitively, specifically for the stories with action verbs in them, and only on the questions relating to those action verbs did these PD patients perform worse than the healthy controls. That sounded pretty cool. Especially because I reviewed the paper, so I thought this is really exciting. This we have to do in Danish. So Marie-Louise and Sabine fortunately said, yes, that's a really good idea, let's do that for a bachelor's thesis. And they created these stories, very nicely controlled with all sort of linguistic factors and the same syntactic structure crossing over when you had action content in one story and not in the other, and so on and so on. And I think we called in at least 30 PD patients or more and healthy controls and tested them. And you can almost hear the excitement in my voice, showing no effects whatsoever in Danish. Right. Of course, we also did some patient analysis on it, and we don't have enough sort of patients to say that there is also a strong null effect. There's just some murkiness over it, but definitely not a very strong result. Fortunately, we also had a senior author on this paper who then said, who were those people reviewing this original study? And then I had to say, I at least know one of them. And then if you go back and look at this, you can actually see that for the three other tasks, you have pretty clear ceiling effects, especially for the controls, which you don't have as much here. So this is most likely just a slightly more difficult task, where you can get maybe some of that very subtle cognitive impairment in the PDs that are there, right? Something with memory, something related to that or whatnot. Julian?
How do you measure the degree of normal functioning?
With the test that Trump proudly said that he passed, namely the MOCA, the Montreal Cognitive Assessment Task. So if you pass that, then at least you're not mildly cognitively impaired. That's something, I guess. It's a pretty neat task where you can score a maximum of 30. And if you get less than 26, it depends a bit on what country do you do it in, then you are mildly cognitively impaired, and then below 20, it's even worse. So it's a lot with memory. There's a verbal fluency test in it. You have to produce some drawings. It's just sort of it's a very condensed cognitive battery task kind of thing. Yeah. Okay. So luckily we had the senior author on board who could direct us to this, and that tells us at least that there's some sense in this story, and that this is probably a little just of a fortunate result in this case. Yeah. So the jury is definitely still out on whether or not action language per se is affected in PD patients. Yeah. And as I said, I did not expect us to get to response inhibition. So I will only sort of end on saying, mentioning that that's the other aspect of what we do with these PD patients, namely that in the clinic there are many reports from the spouses on increased impulsivity after especially deep brain stimulation. And what do you make of that? Well, again, impulsivity, like verbal fluency, is not the easiest thing to disentangle. So what we're looking at for now is response inhibition. And then we've sort of wavered a bit between proactive and what we call reactive response inhibition. So the proactive inhibition is the one measured by the go-no-go task and is a bit more sort of you're remembering that you can't do a certain thing and so on. Whereas reactive inhibition is stopping something, an unwanted action. So that's what we measure with the stop signal task. And that task is a neat one because you get some pretty sort of clear behavioral measurements that we can also very directly relate to their brain signals. Hopefully looking at the sort of response, the inhibitory processes, trying to disentangle it under different stimulation conditions. Yeah. That's it for me. A huge thank to both Erik and Mikkel Petersen on the direct neural help. Mikkel was the senior author who made sure we looked an extra time at the results.
South American.
The South American group's study. Yes, please, this is recorded, so you know we have you on tape. Furthermore, Mikkel also made a really good point about this action content, namely that if we look at the stories, it's not sort of like action heavy. Also, you could discuss, is it only the verbs that constitute the action, or is it the whole event? And when do you sort of ask about circumstances from an action event, and then it's only circumstances and so on. So there are many things to unravel further if one wants to go this way. Luckily, we have a null result, so it's definitely a promising avenue to further pursue. Good. And then Marie-Louise and Sabine are the ones doing all the work on that one. And Aira is doing a PhD in Norway now, in Oslo, so hopefully I can nab her next semester and see if we can do both a Norwegian and a Danish explicit semantic analysis. Yes, that's it for me. Thank you.